Niacin's anti-inflammatory potential, especially in the form of niacinamide, offers a compelling yet largely overlooked avenue for supporting joint health and managing chronic inflammatory conditions like osteoarthritis and rheumatoid arthritis. While most of the public associates vitamin B3 with energy metabolism or cholesterol management, its impact on inflammatory pathways and immune modulation suggests a deeper therapeutic potential, particularly for people suffering from joint pain, stiffness, or autoimmune-driven tissue damage. The story of niacinamide and joint mobility illustrates how a low-cost, well-tolerated nutrient can quietly offer relief in areas now dominated by expensive, high-tech biologic treatments. Niacinamide, the non-flushing form of vitamin B3, has been shown to suppress key inflammatory mediators such as C-reactive protein, CRP, and tumor necrosis factor alpha, TNFA. Both of these markers are well-established contributors to joint inflammation and cartilage degradation. Elevated levels of TNFA in particular are associated with the progression of rheumatoid arthritis, where the immune system mistakenly attacks joint linings. Niacinamide appears to help regulate this immune overreaction, potentially reducing pain and preserving joint structure. Additionally, it influences leukocyte migration and cytokine production, making it a subtle but comprehensive modulator of the inflammatory cascade. Clinical research into this effect goes back several decades, with some of the most striking studies emerging in the 1990s. A randomized, double-blind study conducted by Dr. William Kaufman and later echoed in follow-up trials found that high-dose niacinamide, often in the range of 500 milligrams to 3,000 milligrams daily, significantly improved joint mobility, reduced swelling, and lowered the need for NSAIDs in osteoarthritis patients. Participants experienced improved range of motion in affected joints, as well as a decrease in subjective pain, allowing them to reduce or eliminate common painkillers, without the gastrointestinal side effects typically associated with long-term NSAID use. These results, though modest by today's standards, suggested a nutrient-based way to modify disease progression, not just mask symptoms. However, the rise of biologic therapies, engineered monoclonal antibodies targeting cytokines like TNFA or interleukin-6, has largely eclipsed interest in niacinamide's use in inflammatory arthritis. These drugs, while highly effective for many, are expensive, can suppress the immune system broadly, and often require injections or infusions. In contrast, niacinamide is inexpensive, orally administered, and generally well tolerated at therapeutic doses, making it accessible even to patients in lower resource settings. Yet it rarely features in modern treatment algorithms, partly because it lacks the commercial momentum that drives newer drugs through clinical trials and into clinical guidelines. Moreover, the shift toward pharmaceutical interventions has perhaps caused a kind of therapeutic amnesia, where earlier findings on vitamins and inflammation have been relegated to the past, dismissed as outdated or insufficiently rigorous. This is unfortunate, because the mechanisms by which niacinamide modulates inflammation are still biologically relevant and now better understood thanks to modern immunology and molecular biology. It influences sirtuins, PARPs, and NF-B signaling pathways, all of which are implicated in inflammation, oxidative stress, and cellular repair processes that affect joint tissue health. The issue is not whether niacinamide should replace biologics or NSAIDs. Clearly, in severe cases, those therapies are life-changing. Rather, the question is why niacinamide isn't being more seriously considered as part of a multi-pronged approach to joint inflammation, especially in the early or moderate stages of disease. Its low-risk profile and systemic benefits, including support for brain function, DNA repair, and cardiovascular health, make it a rare candidate for wide-reaching, integrative care. It could also be of particular benefit to individuals who are sensitive to NSAIDs, are looking for adjunctive therapies to reduce medication load, or who prefer nutritional approaches when possible. One of the challenges to niacinamide's mainstream adoption in this space is dose. The anti-inflammatory effects typically require higher doses than those found in multivitamins or standard B-complex supplements. While well tolerated by most people, these doses are often outside the range considered nutritional and fall into the realm of therapeutic application, where clinical oversight and individualized monitoring become more important. But this distinction shouldn't be a barrier. It's a reason for renewed investigation and smarter use, not dismissal. In a broader sense, the niacinamide-joint connection underscores a recurring theme in modern medicine: that some of the most useful interventions may be the ones that are inexpensive, underpromoted, and biologically grounded in the body's own regulatory systems. While the healthcare world continues to focus on targeted, high-tech interventions, there remains a strong case for revisiting foundational compounds like niacinamide, particularly when they show potential in conditions that affect millions and strain both quality of life and healthcare resources. Ultimately, niacinamide's ability to improve joint mobility and reduce inflammation deserves a second look. Not just as a historical curiosity, but as a practical, evidence-informed tool that could still play a meaningful role in personalized, integrative approaches to chronic joint disease.